All right. Hi, everybody. How you doing? I hope that.
Fine, thanks.
Good job, Siri. Good job. Um, I twisted the heck out of my ankle, so I'm going to sit while I teach today instead of hobble. Um, so today I'm going to be talking about some additional information about barrier immunity. These slides are a continuation of Wednesday's slides, so there is not a second set of slides posted. Also, please remember the clinical assignment that is posted on Moodle and due next week. So be aware of that. Basically, there are 3 cases to read about, and each case has 3 questions. And it's the same 3 questions for all 3 cases. And if you have questions or want to talk about things, I am more than happy to talk about things. So on Wednesday, let's move ahead to kind of stuff we talked about on Wednesday. One of the things we talked about on Wednesday, or sort of the area that we got into on Wednesday, was this idea of barrier immunity and thinking about the immune responses at barrier tissues. All of these tissues are sort of unique in that they are constantly in contact with microorganisms and are unique anatomically because they are trying to prevent microbial infection. But you might imagine they also need to be somewhat unique immunologically because if they were making inflammatory responses or immune responses 24/7, that might not be something that made you feel great. One feature of all of these tissues that we talked about last time is that they all have a group of commensal microbes that provide a number of different benefits to you, including providing colonization resistance so that other pathogenic organisms don't have a niche to come into. And so that's actually one layer of defense at all of the barrier tissues. We also talked a little bit about how we can divide up the barrier tissues, and I gave you sort of the simple definition of things that are kind of wet versus things that are not, in terms of the skin versus other mucosal sites. For the rest of the time today, I'm going to be focusing on the other mucosal sites. There are plenty of really fascinating things about skin immunology, particularly how things like wound healing of the skin is interrelated with immune responses. There are some really cool papers that, and I will admit that one of them is from an alum of my lab. This was her PhD work, showing how hair growth, having your hair regrow is related to immune responses in the skin. So there's all sorts of cool stuff with that. But most of the time, when people are thinking about mucosal tissues, they are thinking about one specific tissue. So these are sort of the mucosal tissues. Most of the time immunologists think about barrier organs, they are thinking about mucosal tissues. And so we're going to specifically, instead of just thinking about barrier tissue, zoom in on mucosal immune responses. And you can see the mucosal organs here. They include parts of the respiratory tract, like the sinuses, the trachea, the lungs, parts of the GI tract, the oral cavity, esophagus, stomach, pancreas, intestine, the conjunctiva, so the eyes, the lacrimal gland, which is where tears come from, the salivary gland. You can also see the urogenital tract, as well as a lactating mammary gland, which is also known as a mucosal tissue. And I absolutely really liked the way that your textbook described what mucosal tissues were and what was important about them. So I quoted it here, which was to say that all of these tissues are sites of communication where material and information are passed between the body and its environment. And so if you think about that, we think of these tissues as being a barrier to keep out microbes, but they also have to be able to let stuff in. As you can see, there is this material and information passage there. And so you can't just have a gigantic wall. I mean, the skin is a gigantic wall in a lot of ways. But these have to be able to let stuff in. So you can see, because of their functions of gas exchange, food absorption, sensory activity, and reproduction, they're by necessity dynamic. So they've got to be able to change quickly. They have to be relatively thin, and they have to be relatively permeable. And how in the world you defend in those kind of areas can be a real challenge. And they all have vital functions, and they're also sometimes a bit fragile because they're thin and they have to let things through. And so that has meant that these tissues tend to have some really specific unique organization and unique aspects of defense. We've already talked about one aspect of a unique response at mucosal tissues earlier this semester. And that is secretory IgA. So if you remember when we talked about the different isotypes of antibodies, we talked about how IgA has a form. It is the major form that people study. So in fact, I didn't even tell you really that IgA could do anything else. It can, but this is the main one that people think about, is that IgA is secreted. I might be able to do this, possibly, maybe. Yeah. Oh, yes, I can do this. Laser pointer, that's what I want. So here we've got a mucosal tissue. This is the surface that's sort of in contact with the outside world. Here is the inside part of the body. And what you can see is that our IgA is actually in that mucosal tissue. It's been secreted into the mucosal tissue where we can neutralize pathogens or toxins before they get in. And so one of the unique features of the mucosal tissue is they have secretory IgA as a line of defense to neutralize microbes that are there. Secretory IgA can also be used to help export microbes. So we can see if a microbe needs to be exported or a toxin needs to be exported, you can see this here. As secretory IgA is getting secreted, it can take stuff out with it and bring those outside. So secretory IgA is one of the unique features of the mucosal immune system. There's another really important feature of the mucosal immune system that we can think about very broadly. And this one is obvious or implied in the name of the mucosal immune system, which is that we also have a whole lot of mucus in this location, which I am sure you are familiar with in some form. So mucus is basically a polymer where there is this one little protein that gets a ton of glycosylation, and then when that one little protein is outside of cells, it polymerizes to make this super giant molecule. That's some proteins that are making almost like a scaffold that are just covered with sugar. And so you can see all of this covering. It's basically a layer of goo. The amount of water that's there can change to change how thick or thin that mucus is. And basically that mucus, again, you are probably familiar with mucus in some way or another, is a layer that sits outside of the cells so that there is a bit of a barrier between the cells and any particular microbe. So this is showing different parts of the intestine, the small intestine and the large intestine. And in the gray, you can see the cells and their absorptive capacity. And then in the green, you can actually see how much mucus sits on top of those cells, how much of this basically sugar, big polymer, sits on top of those cells. Notice here in the duodenum, it's about 100 micrometers thick. That's almost the same thickness as the cell layer. And so if a pathogen wanted to interact with or one of these cells for infection, it would have to get through this whole sugary goop layer. And you can see the specific details of those layers changing at different parts of the GI tract. But in any part of your mucosal immune system, you are going to have that layer of mucus helping to protect the cells from infection. You can see this here as well. So here is our layer of cells. We've got the immune cells underneath, inside the tissue. And outside the tissue, we have our secreted IgA, as well as all of our mucus, giving us a nice layer of protection of the barrier. You can also see this in this other figure. I liked this figure just because here you can see the cell layer, and you can see all of this mucosal layer, or this mucus layer, outside of the cells. And you can hopefully appreciate the scale of all that mucus layer compared to the cells, and the ways that all of these different microbes can get caught in that layer and not penetrate all the way down into those cells. So it's not a layer of dead cells like the skin has, but it is some kind of physical blockade that is helping get rid of some of those microbes. And we know about specific cells called goblet cells, whose job it is to just secrete mucus. We also know that in mice who have a certain, I guess a better way of doing it is if you first look at this image, this microscopy image, the blue cells, the blue staining, show you the cells of the intestine, and the green staining show you the microbes. And what you can see is there's a big gap where the microbes can't touch the cells of the intestine. And that gap, this empty looking place, this black part, is where all the mucus is, keeping these two apart. There are some situations where there will be mutations so that that mucus layer is gone. And so you can see in that situation, the microbes are now going to be in contact with that barrier. And so at any part of the mucosal immune system, one of the things that we have protecting us is the mucus. These are not the only unique features of the mucosal immune system. So when we think about the mucosal immune system, some of the things that are unique have to do with anatomy. And in some ways, I would say that the mucus is kind of an anatomy thing. The presence of secretory IgA, you can argue for yourself if that's anatomy or an effector function. It's not clearly labeled as either one here, and I think that 6 of one, half dozen of the other could be some of either. But there are also some other types of anatomical features that we'll see, as well as some different types of effector mechanisms and some other unique features of the environment. And I'm going to show you some examples of each of these things in a mucosal immune response. As I mentioned a little bit ago, when immunologists study the mucosal immune system, if you look, for example, in your textbook, this chapter 10, is called the mucosal immune system. In some other immunology texts, this chapter is called the barrier immune system. It's because those texts have a little section on skin outside of mucosal immunity. But if you actually took chapter 10 of your book and looked at it really closely, you would see that when it is talking about the mucosal immune system, it pretty much always uses the same anatomic location as its example. And when most people think about mucosal immunity, they think about one particular mucosal immune organ instead of talking about what's up with all of the mucosal immune organs. For a long time, we have really lumped them all together. Just all the mucosal immune system works the same way. I think that we are starting to understand that that's not totally true. In fact, one thing that I learned during COVID is that the upper respiratory, like the nose immune system and the deep lung immune system are completely different. But your textbook just says they're all mucosal immunity. And so we're going to be talking about kind of the organ that everybody thinks about with the mucosal immune system. So if you were looking quickly at chapter 10, you would really think it was about one organ, not the whole mucosal immune system. One reason why I laugh about this is I actually was on FaceTime with my sister last night. My sister did her PhD on B-cell responses in the lung, and she thinks about mucosal immunity a lot. And I told her about what I was talking about today, which is the famous mucosal organ, which is not the lung. And she spent a lot of time being like, oh, how would you leave out the lung? She's very angry. So note that this is sort of the way your textbook calls the mucosal immune system, but we are really picking one organ that we're focusing on. And that one organ is the GI tract, specifically the intestine. That's the one that everybody talks about when they think about mucosal immunity. So know that we probably are leaving out a lot of things that go on in the mucosal immune system in some of these other organs. I have another friend who works specifically on the immune system in the mammary gland. And there are many ways that what she studies is unique from what we're going to see today. But we're going to go big picture instead of zooming in on some of those little details. So we can look at some of our sites in the mucosal immune system. And one of the things that we see is on that list of anatomical changes. And so in any of our mucosal tissues, we actually find that there are organized lymphoid structures. So it's sort of like in each of these mucosal tissues, there are like mini lymph nodes to help with the surveillance because there is so much encounter with microbes going on. And so here we can see the wall of the GI tract. You can see we've got the extended epithelium to give us lots of surface area for absorption. So here's our part of the intestine that is absorbing nutrients. But you can also see that there are other little areas where we have these little lymphoid tissues that kind of that are officially secondary lymphoid organs and look like mini lymph nodes. And you can also see that we have lymphatics going through all of this tissue. So one unique anatomical feature of our mucosal sites is they all have these organized lymphoid tissues. You can, in fact, see this in cross-section. You can even see some germinal centers in some of these little lymphoid follicles. Some of them are so big, they get special names like the Peyer's patch. Sometimes there are just little lymphoid follicles that form in places as well. As is also implied on this slide, there are some unique types of cells in some of these places. And one of those particular cells that's really important is called an M cell. An M cell stands for a microfold cell. And you can see an M cell in the wall of the intestine in an electron micrograph on the left. So what you should notice here is the area of the intestine where the M cell is looks different. M cells would also look different if we were somehow able to do our electron microscopy of the bottom and look at them from the inside. And this, of course, since that would be awfully hard to do, we instead have a cartoon. And so here you can see the M cell in the lighter pink compared to the other cells of the intestine in the darker pink. And you can see that the M cell is shaped funny. and basically makes this little pocket internally. And that's a little pocket where immune cells can come hang out and take a look at antigens. The M cells can capture microbes and bring them in just in sort of small numbers to show them to B cells and T cells so that we can start to get a response. And so it's sort of like, instead of just letting in all of the microbes, instead of having an open wall where you just let the microbes come into the gut tissue, the M cell takes little samples and brings them into this secluded little area and says, hey, T cells and B cells, can you check these, see if they're okay? We're not going to actually let them all in because they might be bad, and it would be really terrible if we left them all in. But we're going to bring a few in. We're going to keep them in this secluded special zone. We're not going to actually let them go through the whole body, and we're going to let you look at them. I'm sure we can come up with some, like when they have the special screening area in the airport or something, the like special little zone where they bring things to check it out. And this is all the job of the M cells, the things that will happen in this little pocket, and this sampling that the M cell is doing. We can also see that there are actually dendritic cells that can reach between the cells of the intestinal wall to try to capture antigens outside. You can see this in a cartoon in the left, where you can see this yellow dendritic cell reaching in between the cells of the GI tract in order to phagocytose things that are actually in the GI lumen. And you can also see this in the microscopy image on the right. The black area is the GI lumen, the green is some dendritic cell, and you can see that dendritic cell reaching up really and trying to go on the other side of the cell barrier in order to sample. So these are some of the important anatomic things going on here. There are a couple of other important immunologic things as well. And one of them, to be honest with you, I would say that the next 3 slides are probably some of the things that are sort of, if you ask me, like, what is the most important thing, or if you had to say one sentence about the mucosal immune system, it would be related to the stuff that's on the next 3 slides, because this is just, to me, like the thing that's super special about the mucosal immune system. In the top part of this slide, you actually see an image that's pretty similar to an image you've seen earlier in the semester, where you can see some kind of tissue. Here we're saying it's the skin. I'm not going to deal with exactly which tissue and whether I think the skin is the best tissue. Maybe it is the tissue in, well, no, I won't go there. It's some kind of tissue. You can see that tissue has some kind of macrophage that lives locally in the tissue. You can see that something happens so that there are bacteria that come in, and that local cell starts an inflammatory response. And we get swelling, and we get vasodilation, and we get increased vascular permeability, and we get our whole classic inflammatory response. This is the inflammatory response that we've all talked about. And again, the top of this is basically the same figure you've already seen. If you looked at the same process in a mucosal tissue, you would see something different happening. There are still innate immune cells like macrophages that are living there. So here's a macrophage living in the mucosal tissue, just like a macrophage that we saw living in the skin. That macrophage can sense some kind of microbe. But when that macrophage senses that microbe, that macrophage won't induce the same kind of inflammatory cytokines and won't induce an inflammatory response. And so the response that we see will look pretty different than the response that we see in the mucosal tissue. A large part of that is because of sort of different cytokines and different things coming from that original innate immune cell. So here we can see some of those innate immune cells either responding in the M cell pocket or doing some phagocytosis. They're not going to lead to a traditional inflammatory response. Those innate immune cells that live locally in this location are a bit different. And they are often referred to as cells that have this sort of immunoregulatory ability. And when I was putting this slide and the next couple of slides together, I sort of was snickering to myself a little bit because I wrote the immunoregulatory environment. And then I remembered that immunoregulatory is immunologist code, and that I probably was going to have to totally define for you what immunoregulatory means, or at least what an immunologist means when they say immunoregulatory. I mentioned this a little bit in One Day in the Past, and I'm not going to go through the whole story now, but once upon a time, there was this idea of T cells that could turn off other T cells. They were called suppressor T cells. And then for various reasons, people stopped believing suppressor T cells existed. It's a whole big thing. And if you asked immunologists in that time, they would be like, what? Suppressor T cells? I never said there were suppressor T cells. I don't know what you're talking about. I don't believe in that. No. And in fact, when I was an undergraduate, I was kind of taught that the word suppressor was a dirty word. We do not use the word suppression or suppress or suppressor. Later on, we found some T cells that turned some other T cells off. But at this point, the word suppressor or suppression or suppress was still thought of as a dirty word. You couldn't use that word. That word is bad. And so instead, we called the T cells that did the off stuff regulatory T cells, because we didn't want to use this sort of bad word. And so immunologists have kind of taken on the idea of using regulatory to mean off. It's like it's our code for suppression. I think when a lot of people think about such and such is regulatory, they think they mean like controls or something like that. Immunologists have a little bit more, they mean a little bit more almost towards off or suppression when they say regulatory. And it's kind of because the idea of suppression got a bad name once upon a time. And so we say that the environment in mucosal tissues is immunoregulatory. What that means is that those innate immune cells, like this macrophage that we see in the mucosal immune tissue, like the GI tract, is going to make responses that are not really inflammatory responses, that are instead responses that might lead towards more Tregs and other kind of suppressive types of responses. So this innate immune cell in the gut might specifically make cytokines to push T cells towards Tregs. That might make a whole bunch of TGF-beta and IL-10, whether it's a macrophage or whether it's a dendritic cell in that location. You can sort of imagine that all of the innate immune cells in the GI tract know that they're in the GI tract. And when they say when they see antigen, their response is sort of to be like, everybody calm down. While in systemic tissue, we start this inflammatory response and we ring the alarm bells. Instead, those same innate immune cells know that they live in the GI tract or in a mucosal tissue, and they produce a different set of cytokines to sort of keep an immune response from getting too high. This can be actually a number of different molecules that are being made from those innate immune cells. We now actually think that some of the microbial products of our commensals help to regulate this, and so some particular fatty acids or vitamins that are made by the commensals can even further push us towards an immunoregulatory environment. So let's think about this in terms of the good side, because there's a good side to this and a bad side to this. So let's think about the good side to this first. Why evolutionarily, or why sort of in general immunology terms, might it be a problem if you had what happens at the top happening in your mucosal tissue, like in the intestine? Why might that be a problem? Yeah, Karamay.
Because on the top layer, like your skin, it grows in size.
Okay.
So like mucosal tissues are usually like within very tight spaces.
Okay.
I'm thinking in terms of like nose, so like in case
Okay.
if something like that happens, like it will swell up.
Okay. So one of them is kind of a space problem of whether or not you have room to have that inflammation. And that is certainly related to kind of what I was thinking about. I hadn't thought about it that specifically. I thought about it more broadly, but that's totally like in that direction. What? Yeah, Michael.
Since the mucosal tissues are interacting with microbes all the time,
Mm-hmm.
like if it made the inflammatory response, it basically be inflamed constantly.
Yeah, it basically, since we have all of these microbes, you can't, like, you don't live in a sterile box. Your mucosal tissue is 24/7 in contact with microbes. If your mucosal tissue was inflamed 24/7, you would not feel good. And in fact, you would probably have a fair amount of tissue damage. You would also have blockade of the function of those tissues, like you could imagine with the space constraint that Karamay mentioned. You would actually not be able to have your nose work correctly if it was always full of swelling and it was full of inflammation. If your lungs were 24/7 full of inflammation, they probably wouldn't be able to do gas exchange very well. If your GI tract was always in an inflamed state, it probably wouldn't be doing food absorption very well. And so, in fact, these are all organs that are incredibly important for you, for your normal physiology. And if there was tissue damage, you'd be in bad shape. And if there was excess inflammation, you would actually stop the function of those organs. So one problem is you need to make sure that you don't have any kind of excess inflammation in those organs. Because if you did, you'd be in bad situation. So instead, the innate immune cells are sort of saying, everybody calm down. We're in a location where we cannot have an excess type of response. If you have inflammation in your intestine all the time, that is called Crohn's disease or inflammatory bowel disease or colitis. That's what's happening to you, is that you have too much inflammation all the time in that location. Your immunoregulatory environment is not kind of working out. There's one other reason why we don't want to have an immune response being made all the time in the GI tract. So let's think about this a little bit more kind of big picture for a second. Big picture immunology is you respond to organisms that are foreign, right? You respond to organisms that are not you. I'm going to name some organisms that are different than me. That are foreign from me right now. Okay? Some organisms that I am not include a cow, a salmon, wheat, rice, oranges. I'm trying to think what I ate today. I had orange juice and I had cereal and milk. Last night I had some salmon for dinner. None of those things are me, right? It would be really bad if I made immune responses to all of those other organisms, right? My GI tract is constantly encountering foreign stuff. Even if it was totally sterile, yeah, I wouldn't have PRRs, but I'd still have some antigens that my B cells and T cells had never seen before, in the case of whatever my food antigens are. And so if we made an immune response to all of our foods, because all of our foods are unique organisms, except for maybe the Diet Coke I drink way too much of, that, again, would be really bad evolutionarily. We would not survive or reproduce particularly well. And so what we've actually realized is that in mucosal tissues, and this is really well described in the GI tract, we have what's known as mucosal tolerance. And so when an antigen is first encountered, if the first time you ever see that antigen is in the GI tract, then you end up getting a tolerogenic or a Treg type response, and you actually don't make another type of immune response. So when we look at food proteins or commensals, we largely will see Tregs being induced instead of other T cells. And we really see no other response in the system. This was actually one of the ways that Tregs were first described. One of the first sort of places where people were like, oh, right, these suppressing things kind of do exist, was in understanding Tregs. Tregs were originally called Th3s before we understood a lot more about what was going on. And we've actually done a lot of experiments where we can take advantage of this. So one experiment that you can do is you can take some mice and you can inject a foreign protein. That foreign protein is called ovalbumin. It comes from a chicken. It's a protein that immunologists love to use as an antigen, so we just use it. It's basically the sample antigen in a lot of immunology. You could give some mice some ovalbumin in an injection. And it would sort of be like a vaccine, where you then looked to see, did those mice make an immune response to ovalbumin? And they would make a huge immune response to ovalbumin. Again, it would be like you basically gave the mice a vaccine against ovalbumin. So if ever they were attacked by chicken proteins, they would be safe. You can just make it, it's a foreign protein, you inject it in, you make an immune response. If, however, before you do that, you feed the mice some ovalbumin, so that the first time the mice have encountered this antigen, they are encountering it in the GI tract, and then you immunize with ovalbumin, you actually don't get much of a response. Because that initial immune response in the GI tract drove Tregs and made this much more immunoregulatory response. This is something that we take advantage of in certain situations with some allergies, in terms of trying to give certain antigens in the GI tract to try to push you to have some Tregs. this is hypothesized to be involved in increasing numbers of peanut allergies, in that a lot of little kids are, in the past, the first time you saw peanuts as an antigen, you saw them from eating peanuts. More and more, the first time you see peanuts as an antigen, you're seeing lotion that has peanut oil in it. And so perhaps you're getting a slightly different response. That's very much hypothesized for a lot of what's going on with peanut allergy. So your first exposure gave you yes response instead of regulatory response. Sometimes people ask me, this is a question that I get asked pretty frequently, and I want you guys to answer this question, because I get asked this question a lot when I'm giving talks to the lay public. I'm scared of needles. I don't like needles. I want you to give me all my vaccines as pills that I take orally. What do I usually say? Or what sort of is this something to be aware of in response to that? Yeah, Jay?
I mean, if we take the pills, the first response that we have for that antigen will be through the GI tract, and so we'll be getting, like the next time when, in the same case like the mouse, next time you get like the antigen, you get a self-response.
Yeah,
So
sometimes if you see an antigen for the first time in the GI tract, you're going to get a tolerance response. You're going to make Tregs. So it's like that vaccine is actually going to make your life worse, not better, if we give it through the mouth, as opposed to giving it systemically as a shot. Obviously, there are some exceptions, and there are some ways that we're learning about these responses to try to figure out how we can start to sort of modulate them and work with them. There are lots of ways that working with them could be really important in thinking about how in the world we deal with allergy, but also how we could sort of do some of these vaccinations. But one of the things I tell people is right now, a lot of what we know about your GI immune response is about how you actually turn off immune responses when you see something in the GI tract. The reason why you actually make sure that you don't make immune responses to cows and can eat a steak. And so if I'm immunizing you in the GI tract with a pill, well, yes, it might be less scary, could actually immunologically be really scary, because it could actually predispose you to worse disease, not better. Again, there are a few examples out there of oral vaccines, but they are the vast minority, and there are some special things going on in every case of an oral vaccine. So you should realize mucosal tolerance is quite important. This also sometimes leads to one other detail. So we've talked about sort of the good side of mucosal tolerance, but what about the potential bad side? So do you have any questions or any sort of things that you look at here and say, wait a minute, there's still a piece missing. We still got a problem here. Yep, Christina.
How do you deal with like bugs in the intestine?
Yeah, sometimes you actually do get infected with a pathogen in the intestine. So what the heck happens there? Look, Christina, you even had the question on the next slide. So we do kind of have to think about, okay, there are actually some pathogens that are going on here. Some of this will be a little bit of something that we deal with anatomically. So you think of the GI tract. You think about how the GI tract works. Nothing that's in the GI tract stays there forever. You are eventually forcing everything out at some point. And so part of it is that, we can have changes in how much we have things leaving in order to try to flush out potential pathogens. But we can also sometimes see a mucosal immune response to pathogens. And it is, in fact, important that we have this mucosal immune response to pathogens. And so we will sometimes, see a sort of more typical systemic response in response to pathogens. And this table from another immunology textbook, the Janeway immunology textbook, I think gets at this distinction really nicely. So sometimes in the mucosal situation, we get mucosal tolerance. In other times, we do get sort of the more traditional immune response, which is they're calling here protective immunity. And they make a nice distinction in this table. If you look at this table, particularly with regard to antigen, so I would say that the difference is in the antigen, and because the antigens are different, the response is different. But if you look at what this table says about the antigen, what's different about the antigens in these two situations? Yeah, Michael.
In the mucosal tolerance, it's like antigens that would just stay in the GI tract, while in the protective immunity, it's like antigens that would like try to invade the cells.
Yeah, notice that this says invasive bacteria. And realize that viruses, again, are, as Michael says, are going to invade and get into other parts of the body. Toxins are going to get in. And so the difference is seems to be based on sort of whether or not the antigen seems to be trying to invade, whether or not there is invasiveness going on here. This just shows you a couple of examples, though there are so many examples of different types of microbes that can try to invade. So here you can see organisms, I believe this is trying to show you Shigella, but it could be Shigella, it could be Salmonella, it could be a type of E. coli. There's a bunch of them that will actually try to interact with the cells of the GI tract, and that will actually poke holes in them to try to get nutrients or other things. And so you can see that sort of formation where the cell is actually trying to get stuff out of the cytoplasm of the cell. Here you can see some Shigella that actually force their way in to different types of cells. Here you can see other microbes, Salmonella, forcing its way and trying to invade into other types of cells. So what we might imagine here is that there is this distinction in terms of whether we're going to make an immune response that is more tolerance immune response, more regulatory if something is not invading, and maybe those same innate immune cells are going to actually make the traditional inflammatory response if something tries to invade. And so in the mucosal tissue, this is what we see in the bottom if we're looking at some non-invasive microbe. But this same macrophage, if the microbe was sort of trying to invade, could give us some inflammation. It's just sort of trying to decide, is this an invasive or non-invasive thing? Is this a thing we should be worried about trying to go big picture or not? So I like this table because it talks about invasive things versus other things. So that leads to one question on the side of the innate immune cell. How might an innate immune cell know if a microbe is invasive or not? How in the world is that innate immune cell going to know whether or not a microbe is invasive? Does it have the ability to interview the bacteria and be like, what are your thoughts and feelings? How's it going to know the difference? Yeah, Michael.
We have things like MHC class 1 and 2 that can like differentiate between them.
Okay. So, but remember, MHC class 1 and 2 is mostly going to be about what we present to T cells. And we need to think about whether that innate immune cell is going to make an inflammatory response or not. Is it going to make inflammatory cytokines? So that often is, in some ways, something we can think of being a little bit before or separate than the MHC part. But you're right. You can already see there are some places where the immune system can distinguish inside versus outside. So how might a innate immune cell distinguish a invasive versus a non-invasive microbe? Yep, Karame.
Would it have to do with like its receptors?
Okay, what about the receptors?
They would be, what's it called? It would be easy to detect if one of microbes because they attach to it.
Yeah, what if you took receptors and you had some of them on the cell surface? Those would detect things that were outside, but not things that were inside your cell. And what if you had some other receptors that were inside the cell? A microbe that was not trying to invade would touch the ones on the outside of the cell, but not the ones on the inside of the cell, because it didn't try to get in. A microbe that was invasive would be hitting both those outside receptors, but it would also hit the inside receptors. So you could imagine a way that we could distinguish the invasive microbe versus not, based on, do we just detect it outside of cells, or do we start to detect its products inside of cells too? Oh my gosh. Way back in the first couple weeks of class, when we talked about PRRs, I told you that some were on the cell surface and some were inside of cells. This is showing you that with toll-like receptors as an example, but there are others that are inside of cells, like some of the different groups I told you about previously. So right now we think that at least part of this has to do with whether the PRRs on the cell surface get triggered versus whether PRRs inside the cell get triggered in that macrophage in the GI tract. So that macrophage can start to figure out, was that thing invading or was that thing just chilling? There are also big differences in terms of other things like genetics and other issues that may change the amount of mucus, the thickness of the mucus layer, in terms of whether or not those microbes can actually start to interact with those cells. And so one of the things you'll notice is that one of the proteins that is involved in mucus is MyD88, which is part of toll-like receptor signaling. And so the PRRs may be helping to control what's going on with the mucus, but also helping to allow the cell to know, is this an invasive organism, or is this an organism that is less problematic? And then, of course, we have the anatomical specializations to just flush stuff out. When we have a microbe that is causing a problem, we also can think about how exactly we deal with those microbes. So how do we deal with the fact that your GI tract is very likely to be in contact with pathogens from time to time, since it's in contact with microbes? As we said, it would be pretty bad if you had damage to your GI tract, because then you couldn't absorb food, and then you would not feel good. Or as Dunaway would say, then you get dead, right? So if you actually had a pathogen there, that would be bad. You don't want to have so much inflammation there. You also don't want to let that pathogen go for a really long time. And so one of the things that will happen when you try to make an adaptive response is that we might cut out some of the steps. that we talked about with the traditional adaptive response. Remember how we talked about what things take so long in making an adaptive immune response? In the mucosal tissues, we cut out some of those steps to try to make that response not take as long and try to get rid of the microbes quicker in the situation where we need to get rid of those microbes. One of the big things that took so long, one of the events that was part of our delay, was trafficking. Trying to move all the stuff to the lymph nodes and to the lymphoid organ, right? Well, that's why anatomically we put lymphoid tissue there. So we don't really have to move things as far. We have a lymphoid organ right there. The other thing that we do is that if a T cell first sees its antigen in a mucosal site, so if, say, we have a T cell, it sees some antigen, maybe first in a secondary lymphoid organ that's in a mucosal tissue like a Peyer's patch, that T cell is going to get specific types of trafficking molecules on its surface. We most frequently talk about a specific integrin, though there are, of course, chemokine receptors and selectins that are important here as well. And that T cell or B cell is going to say, you know what, I'm not going to circulate through the whole body. I think I'm just going to live in the intestine. So that if my bacteria ever comes back, we don't have to worry about the timing of moving this T cell back and forth. This T cell is just going to live in the intestine forever, or live by the intestine forever. And so we see a lot of T cells that will specifically traffic to mucosal sites. And if you look at the walls of the tissues in mucosal sites, you see that there are a lot of immune cells that live there. And so we find effector T cells that just hang out in mucosal sites so that we can cut out some of the delays of trafficking and allow those cells to get rid of microbes ASAP if those problematic microbes are there. You can see this red layer is basically the wall of the intestine, the intestinal epithelium. And you can see that there are actually lymphocytes that live in the wall of the intestine, as well as lymphocytes that live sort of in that intestinal tissue. We call many of those cells that live in the wall of the intestine intraepithelial lymphocytes, or IELs. So you can see they are living right in the walls of this tissue. Some of them have weird innate-like functions. Oh, that was not what I wanted to click. Some of them are like ILCs, the innate lymphoid cells. Some of them also have some weird kinds of TCRs, like gamma delta cells or other kinds of weird T cells. And they basically live in this organ, and you can find similar types of T cells in other barrier organs that are ready to get rid of microbes right away, instead of waiting for some of the parts of the delay of the production of an adaptive immune response. You can see the IELs in cartoon form on the left side of this figure, and you can actually see cells in the midst of the intestinal wall in an electron microscopy image as well. IELs are not particularly well understood, and I have seen estimates of things like 5% of your T cells are IELs, and 95% of your T cells are IELs. Both of those I think are probably wrong. Both a little extreme. But in any case, we don't know a lot about them. There's a lot of things we have not studied well about these cells. Why might it be hard to study these cells? Let's imagine that you feel 100% fine in your GI tract today. And we would like to say, well, okay, so what do your IELs look like when you're perfectly, totally, 100% healthy? Because then we could compare that to what they look like in disease, right? So how would we do an experiment to look at your IELs when you are 100% perfectly GI tract healthy? Now, this will work too. How am I going to study these T cells to just have a list of what they look like when you're healthy? Yeah, Jay?
I'm not sure if this is the right way, but you would want to take a tissue sample.
Yeah. The only way you're going to get these cells that live in the wall of the intestine is to actually cut out the wall of the intestine and pull those cells out. It's going to be hard to pull them out, and some of them are going to die. So you're going to get like 3 cells if you do this. You're going to get very few cells. B, if you feel 100% totally fine, would you like to give me some of the wall of your intestine today and have me grind it up in a tube to try to find 3 cells? Does that sound like a great idea? Not really. And so these cells are incredibly hard to work on. It's very invasive if we actually want to get them out of an individual. So we know some stuff about mice because like we can do that experiment on mice a lot easier than getting a person to be like, oh, sure, have my intestine. And in many cases, the times where we get people's intestines are when people are sick and they're willing to give us a biopsy to actually see what's up. And so we know much more about what these look like in, say, in some cases, colitis patients actually have to have part of their intestine removed as part of treating their colitis. Treats their colitis, and then we have this intestine, and we can look at the IELs and see what IELs look like in a colitis patient. But we have a really hard time looking in a regular patient to see what they looks like for comparison. You could imagine this would involve like a colonoscopy, and this would involve like the biopsying and stuff. And do the IELs get changed when you start sticking organs in places? Like you can imagine that this is going to be a really difficult thing to actually study when you start to think about the anatomy of all of this. And this is one of the reasons why, like I said, I've seen super different numbers for even how many of them there are, because it can be so hard to isolate them and to look at what you see in healthy individuals. The one kind of last thing we should just point out is, who cares? Why do we even care about this? Realize that these are many of the places you're getting infected. These are many of the types of microbes that we often think about. Thinking about what's actually going on in terms of immune responses in barrier tissues is in many cases probably pretty physiologic and pretty important for what's really going on if we want to understand health and disease, even though these are very challenging things to study and things that we are only now realizing that maybe we shouldn't just look at the blood. Maybe we need to look at some of these more challenging to study tissues. Class on Monday is always an interesting place in the semester, or the topic on Monday is a sort of interesting spot in the semester. I think of Monday as our last sort of basic immunology class, and then everything after that as being clinical. Many of the students think of Monday as the first clinical lecture. So Monday is sort of like on the border between clinical and not, because I'm going to talk about peripheral tolerance, which segues us into how peripheral tolerance gets messed up and autoimmunity. So I'm going to largely talk about the peripheral tolerance mechanisms on Monday and then sort of start moving into autoimmunity, and by Wednesday it'll be totally about autoimmunity. Remember, your clinical assignment is due on Monday, and it's on Moodle. Please let me know if you have questions, and I hope that you have a great weekend.